I'm very happy that we started talking about biomarkers, because this is something we have been working on quite a lot recently, but my talk will not be on biomarkers. In fact, it's a bit of a pity. Huh? If I would have known that a few days ago, I would change my topic, definitely, but it's not the case. I'm one of the guys of the second group there that asked more questions than answers. I asked a lot of questions, basically because I hope they come to me because I knew the answer. Huh? So that's basically why I did this. So I'm from Ghent University, and in fact we are part of, here's the pointer. We are also part of different consortia. One of them is also working on intestinal integrity and inflammation in humans and different animal species. And we're also part of 2 different valorization consortia, meaning that we try to develop vaccines and feed additives and food additives for, well, of course for animals, but also for humans. We try to bring these to the market. So this is my topic. As you can see, what we basically do in our laboratory is trying to steer microbial composition using substrates, feed additives, or feed or food on itself, to try to make sure that these microbes produce metabolites that affect host responses, inflammation, metabolites that affect intestinal integrity, but also metabolites that affect the behavior of pathogens. And this is very convenient, because if you ask me questions about nutrition, I can say, okay, I don't know, I'm not a nutritionist. The same here. If you ask me questions about immunology, I have no idea. I'm not an immunologist, and also not really a bacteriologist. So it's fairly convenient. In fact, I'm a molecular biologist, so we try to link all these things with each other, which is a very nice topic. This is what we have seen already before. This is how the normal gut looks like, in small intestine at least. Very long villi, and also, of course, very thin villi. So a lot of absorptive surface, no inflammation, which is basically how the gut should look like in optimal conditions. Now, there are quite a lot of microbes in the gut. So one of the questions is, of course, why don't we react to all these different bacteria that are in quite close connection with this epithelial surface? And basically, I think there are 2 different answers to it. First of all, when animals or we are born, we present a lot of antigens coming from bacteria to our immune system, and we just tell our immune system that we do not have to react against these antigens. That is called oral tolerance. And a second reason basically is that if you look at the epithelial layer, you have a lot of receptors there that are basically responding to microbial components. These are called toll-like receptors, and these are basically found at the basolateral surface, so not at the apical compartment, meaning that once you have a breach in this epithelial layer, when you have intestinal integrity increases, when you have permeability increases, then these bacteria can just go through the epithelial lining, connect with these receptors, and that's really a trigger for inflammation, okay? And that's, of course, what many pathogens do. And if you think about poultry, the poultry situation, you have quite a lot of triggers that induce this kind of inflammation, this kind of permeability increases. Here I just listed a few of them. Viruses, of course, enteroviruses, reoviruses, whatever, but definitely also coccidia do that, also some pathogens, clostridial toxins, even mycotoxins, make sure that there's a breach in this epithelial layer. By killing epithelial cells, of course, bacteria can just penetrate and connect with these receptors and cause inflammation. And inflammation means that there's quite a lot of immune cells that infiltrate in the gut mucosa, and inflammation definitely also means that there's a lot of energy that the animal has to spend to attract these immune cells, so there's a loss in performance. That's basically what this is saying. Now, if you look at intestinal sections of chickens, of broilers, if you look at it using histology, you always see inflammation. The gut is more or less never normal. So there's always something wrong, some minor damage, some subtle damage, but you always see something. So it's never perfectly normal. So as I just said, well, this is the histological slide you have already seen. This is how a section of the same intestinal segment looks like that is inflamed. That means shorter villi, blunted villi, thicker villi, and all these brown dots here, these are immune cells that infiltrate the intestinal wall. That's of course quite energy costly. Huh? And then some other stuff, whatever. Doesn't matter so much, but this is a consequence of intestinal integrity losses. Huh? Now, I'm just going to talk a bit about bacteria, and especially mucosa-associated bacteria. This term has been touched upon already by a few people. These are, of course, bacteria that are very close interaction with epithelial cells, and these are the ones that produce a lot of metabolites that are sensed by these epithelial cells. Huh? And we talked a lot about butyrate in the working group. That's one of them. That's a very important one. That is that is often produced very close to the epithelial cells and also used by epithelial cells. I will come to this back in a few minutes. If you look at the microbial composition in different parts of the intestinal tract, you will see quite some differences, of course, and the most diverse microbial composition you have in the ceca. This is the place, this is the fermentation chamber where a lot of bacteria exist that actually use undigestible stuff, use saccharides also to ferment and to produce short-chain fatty acids, acetate, propionate, butyrate, whatever. There's a lot of different anaerobes, a lot of different families, species, genera present in the ceca. In the small intestine, you only have a few families present and quite a lot of lactobacilli. Huh? So if you sequence microbial composition, if you look at microbial composition in the small intestine, it's full of lactic acids producing bacteria, which is completely different in the cecal compartment. As I just said, well, you have bacteria present in the lumen, quite a lot, and these are the ones that degrade a lot of polysaccharides and other substrates. They produce other compounds, and these are used by other bacteria. It's called cross-feeding. Huh? Bacteria break down certain components, produce intermediates. Other bacteria use these intermediates to produce other intermediates or end metabolites. Huh? And especially important, and this is what I'm going to focus on today, are these bacteria that really are in very close interaction with the epithelial cells. Huh? And there's not a lot known about this, especially not for poultry. Huh? But there are quite a lot of studies being performed in mice, rats, and also humans using biopsies. Huh? And also some studies in poultry. Here's just one of them. We have done some others just using scrapings of the intestinal wall and looking at microbial composition in these mucosa, very close to the epithelial lining. And if you do that, you see that you have, well, only a few, specific bacterial groups being present. And that means in the small intestine, you have some lactic acid bacteria that are very close in interaction with the epithelial layer. Huh? And you have also bacterium with a very weird name. This is Candidatus savagella. It's renamed in the meanwhile, doesn't matter so much. This is a bacterium that is only very recently being cultured, but it's very difficult to culture. But it seems to be very important. I come back to this later. In the ceca, you have butyrate producers that attach very closely to, at least some genera and species of butyrate producers that attach very closely to the epithelium. This is a family of Lachnospiraceae, family of Ruminacoccaceae, and as microbiologists do, you give very fancy and weird names to make sure that people cannot pronounce it. Huh? This is a competitive advantage of a microbiologist, okay? But these ones are really important because they are found very closely to the epithelium. Huh? They produce butyric acid, so they are very important. I will give you some examples why these bacteria are so important in a few slides. Okay? On the other hand, you also find, if you sequence bacteria closely associated with the epithelial cells, so in the mucous layer, you also have other bacteria that you often find there, and these are the more pathogenic ones. Huh? Clostridium perfringens, E. coli, and also in the ceca, a lot of Enterobacteriaceae that also contain E. coli. And as you can imagine, there's some kind of a balance, okay? To be very simple, I would say that if you look at mucosa-associated bacteria and you find a lot of these ones, that means there's some kind of dysbiosis, unfavorable conditions. If you have a lot of these ones, that's favorable. And I will tell you in a few slides that if you do that, and you look at these species there, you will see that it's always like connected with gut health parameters such as villus length, okay? If you have a lot of these ones, you have a high villus length. If you have a lot of these ones, you have a low villus length. And that obviously also is connected with animal performance. But I will give you some data in a few minutes, actually now. So this is the question we have been asking: how is microbiota composition changing when you have an affected intestinal integrity? We have used a lot of different models to try to reduce animal performance and induce intestinal inflammation, and then measure microbial composition, okay? I'm just to give you one example of such a model. This is one of the models we have been using. Basically what we do is we use polydigestible diet, we use coccidial challenge, we use bacterial challenges, and we just a few days later we sample and measure microbial composition. Why we use this kind of model? Well, just basically we want to have an intestinal tract that is inflamed, has poor intestinal integrity, short villus length, etc. So that's ideal, of course, using these triggers. These are just some data, just to show you that this kind of models is actually having the effect we want to see. The challenged animals, of course, have a lower body weight. This is day 26. And these are just some macroscopic scoring systems, either for coccidiosis. This is just a sum of all the different coccidiosis scores. And also dysbiosis scores, which take into account different parameters: inflamed gut, ballooning of the gut, thickness of the intestinal wall, etc. Doesn't matter so much. I just want to show you here that using this model, we see some kind of performance losses, and we also see that the intestinal wall looks abnormal. It doesn't look normal. It looks inflamed, thin, and it also has a lack of tonus. So you just do something that is affecting integrity. Now, again, if you look at histology, you see that there's the villus length is decreased, and that you have this inflamed gut wall. All these cells are CD3 positive T cells. These are T lymphocytes that infiltrate the gut wall, and you see that the percentage of the mucosa, of the wall, of the intestinal wall that is having these immune cells is going up quite enormously using this challenge, and villus length is decreased. So this is the situation we wanted to create, and then we wanted to look at microbial composition shifts. Okay, so basically that's what we were aiming for. We were looking for bacterial taxa, families, genera, species that had a good association with villus length, meaning the lower the villus length, the higher or the lower a certain bacterial group is present in the gut. That's basically what we have been doing. And as you can imagine, well, a lot of bacteria that are potentially located very close to the epithelial cells are found. I will give you the exact data in a few minutes. This one seems to be very important. The higher the villus length, the more of this type of bacteria you find in the small intestine. It has to do with, well, because it attaches to the epithelial cells of the gut, it is more abundant in the ileum. And here you have butyrate producers. So just giving you an idea, and I just go to the real data. This is one of these butyrate producers. In challenge conditions, it's depleted, and also you see that the higher the villus length, the more of these bacteria you find. So this is what you can consider as a bacterial genus that is positively associated with intestinal health. Okay. Also, on the other hand, the more inflammation you have, the less abundant this population is. Okay. So this is Faecalibacterium peronisci, which has been described also in humans to be very important for intestinal health. But also here in this chicken model, and also in other chicken models, using necrotic enteritis or coccidia, whatever, you see that this is positively associated with different gut health parameters. So the more this bacterium is present, the higher the villus length, the more it is present, the less inflamed the intestinal wall is. So it's very important. And this is actually one of the first biomarkers, if you wish, that you can look at, because if you quantitatively analyze the presence of these bacteria in the intestinal, in the fecal material, you actually have some kind of a first test. That's what you could use potentially. This is the second one. I can give you a lot of this information, but this is one that we have been isolating and doing some experiments with. And also here you see a positive association with villus length and a negative association with T-cell infiltration in the intestinal wall. So this is what we can consider as beneficial microbial, microbes that are linked to gut health. Just one slide, I added this 10 minutes ago, just because all of you were talking about biomarkers, at least I plugged one of the slides in. Just to tell you that we have been looking quite a lot the last year on host biomarkers for gut health. And actually we just have patented a set of 9 different proteins that can be used in an ELISA on fecal material just to measure gut health. And this is just one of them. I just give one example, because you really find some proteins in fecal material of chickens that have a disturbed intestinal integrity. This is an example of a protein that is found in heterophilic granulocytes, so in immune cells that go to intestinal wall, but also translocate to the lumen and then release a lot of proteins, okay? One of these proteins has this name, myeloid protein 1. And if you look at it in fecal material of animals with disturbed gut integrity, so with inflammation in the gut, you also see that the lower the villus length, the higher the villus length, the lower the amount of this protein. Or, of course, if you have a damaged gut, you find a lot of these proteins in the fecal material of the animal. The opposite way, if you have an inflamed gut, you find a lot of this protein. So this is something that can potentially be used as biomarker, okay? So we have been identifying quite a lot of these proteins, and I'm quite confident that some of these will be available in the future on an ELISA, on a dipstick, whatever, to be used on fecal material, so that you can just go to a flock and measure this kind of proteins and say, okay, here in this flock you have intestinal inflammation, so we have to do some intervention or not. So this is, or really markers, this is a host biomarker for intestinal inflammation. You also have leakage biomarkers, you also have even epithelial damage biomarkers. You have a whole set of these that will become available. Back to the story. Epithelial integrity, this is something that seems to be very important today. In fact, there are a few different ways how intestinal integrity can be maintained, or how we can repair damage. One of them is, of course, increased cell proliferation. If you have some damage in a villus, some epithelial damage, of course, cells have to renew and double much more. Tight junctions, we have been talking about tight junctions already today. Strengthening of tight junctions is important, and also, of course, anti-inflammatory effects, just because inflammation on itself is also triggering epithelial cell death and loss of tight junctions. Now, as you can imagine, butyrate is having all these 3 effects. I'm not going to give you all the details, but there are a lot of studies showing that butyric acid has these 3 effects on these 3 different important mechanisms for repair of damage. And of course, well, looking at these 2 microbial groups or microbial markers for gut health, these are butyrate producers. And as you remember, these were positively associated with villus length, with animal performance, and negatively associated with inflammation. So it seems to be that these bacteria that closely are located close to the epithelial cells, especially this one, are very key in maintaining intestinal health, because they produce butyrate, which is sensed by the epithelial cells. Going a bit faster now here in this, we have, well, this is a very complex slide, I think nobody can read it, but anyway, we have been isolating a lot of these butyrate producers, strictly anaerobic strains. That's a disadvantage, they're all strictly anaerobic. We have been isolating a lot of these from the chicken intestinal tract, including this Butyricicoccus and this Faecalibacterium prosnitzi. So we have a lot of chicken isolates, and of course then we can wonder, if you deliver these strains to the animals, what is happening? So this is basically a question that we have been asking ourselves. We have been using necrotic enteritis models. You see some data here. This is a study, this is the protocol, it doesn't matter so much. We just induce lesions using Clostridium perfringens challenges. So we induce necrotic enteritis. And if you give this strain here as a probiotic, meaning that we just administer it in freeze-dried form in the feet, okay? Then you actually see that in 4 different independent trials, you reduce the percentage of animals that develop necrotic lesions. So again, telling you that these kind of bacteria are very efficient in counteracting gut damage, okay? And this is just what happens with normal butyrate at different levels. This is also, of course, quite efficient. Just going to, well, this is just showing you that this strain is also having an effect in different dysbiosis models, but I'm not going to focus too much on it. I just want to tell you that if you use models, nutritional models, in which you give poor digestible feed to animals, inducing some performance losses, that also administration of these bacteria stimulate performance. And again, that makes sense if you have listened to the whole story, because these bacteria produce butyrate and also repair subtle gut damage. And actually what this is saying is these are just data from a 16S sequencing study showing that if you administer this kind of bacteria, it really affects the whole microbial composition. So what you don't have is, I mean, you give this bacterium to the animals, it's not only about increasing abundance of this bacterium, but you also increase lactic acid bacteria, and you reduce Enterobacteriaceae, which contain also E. coli and opportunistic pathogenic strains. That's just what this slide is telling you. Okay, just small side story on butyrate and pathogens. We have also been doing quite a lot of studies using butyrate and salmonella, and basically if you use protected butyrate, and I think I'm replying to some of the questions already, if you add protected butyrate, you actually are able to induce increased butyrate concentrations also in the ceca, and you can measure that using HPLC. Depends a bit on the type of compound you use. With some of the compounds, you can really measure it. With a lot of other compounds, you can't, maybe because it's not released, or because it's gradually released, we don't know. But if you do that, you actually are able to reduce colonization with salmonella in the ceca. And salmonella, for example, as a pathogen, it's really colonizing the cecal content. And basically, this is because butyrate is a signal for this pathogen to reduce its virulence gene expression. So this is something that we have been doing a lot of studies on and have been publishing in multiple papers. So it's also a very potent anti-salmonella compound. And that also means that the more of these butyrate-producing bacteria you have, the less Enterobacteriaceae, and also salmonella is one of these Enterobacteriaceae. So that's always some kind of a balance you have, either the other one. So it's one or the other that is most colonizing. Just very briefly, butyrate production in the gut can be stimulated using a variety of different methods. And basically, this is just a general scheme showing you that if you have any polysaccharide, it is converted by some bacteria to smaller components, for example, oligosaccharides, that are used by other strains to produce the short-chain fatty acids, including butyrate. And there are a lot of butyric acid producers that consume lactate. In other words, if you have a lot of lactic acid bacteria, lactic acid on itself, it's in fact toxic. It's not really too beneficial, but it's consumed by a lot of butyrate producers to produce butyrate. That's why a lot of these lactate-producing bacteria, lactobacilli, are very important because they feed butyrate producers to produce butyrate. And as you can see in this graph, you can supplement some oligosaccharides to the feed. These are feed additives, xylo-oligosaccharides, fructo-oligosaccharides, inulin, whatever. And this is also butyrogenic because it really steers this pathway. Okay, I'm not going to give you too much of specific data here, just some examples. This is just a study we have been doing with, showing that enzymes have even a butyrogenic effect. And what we have been doing here was just taking wheat, adding enzymes in a fermenter system, and then just use what is coming out from this digestion to add as a feed additive for broilers. And what you can see is that it increases levels of butyrate-producing bacteria in the ceca, okay? Just showing you that if you have enzymes, okay, you also make sure that you release some components out of polysaccharides, that are out of xylans, of course, arabinoxylans, that are butyrogenic. That's just nothing more than that, but that's, of course, also an effect that is important. It's not only about viscosity reduction, etc. It also stimulates butyrate producers, just addition of enzymes. And here, of course, this is what enzymes do. They degrade polysaccharides, and one of the components coming out, of course, when you add xylanases to the feed, or arabinoxylin oligosaccharides, or maybe if you also remove the arabinose, then you have xylo-oligosaccharides. And just to show you that these components also affect butyric acid production, we did some trials. This is just one of the trials. We did quite a lot of trials with oligosaccharides as substrates. This is just a trial we have been doing with supplementation of xylo-oligosaccharides to the diet of broilers, using a model with poorly digestible substrates without enzymes, okay? Just making sure that the animals perform less. This is a study of a few years ago, actually showing that in this dysbiosis model, because that's basically what it is, we steer microbial composition to an unfavorable one, showing that these xylo-oligosaccharides led to an increase in body weight, decrease in feed conversion, and also stimulate phylus length, okay? But basically what is important is that these substrates, these oligosaccharides, they induce butyrate production in the gut of the animals, because what you see here is a quantification of a bacterial gene, gene copies of this bacterial gene that encodes for this enzyme. Again, it has a difficult name, but this is the final gene. This is the gene that encodes the final step in the butyric acid production pathway in bacteria. Okay, so this is a quantitative PCR showing you that if you add xylo-oligosaccharides to the diet, that the number of gene copies of this very important gene involved in the butyric acid pathway is increased. So you have, this is a measure, in fact, a marker for the butyrate producing capacity of a microbiota of the gut, okay? So this is a test we often use to show butyrogenic activities. So it's an induction of butyric acid production in the gut of the animals. It's a bit of an alternative for measuring butyric acid, just using the pathway. Just some data on the microbes. We often do some 16S sequencing studies looking at the global microbial profile of the gut of these animals. If you add xylo-oligosaccharides, you have an increase in certain lactobacilli. This is just one of them, Lactobacillus crispatus. This is in the hindgut. You also have an increase in some butyrate producers. And actually what happens here, you can read all of it in this paper of Céline de Maaskalk, but what happens is that these guys here, they consume xylo-oligosaccharides and produce lactate, and the lactate is consumed by this bacterium to produce butyrate. So this is a very specific example of this cross-feeding mechanism, and shows you that lactate producers are important because they feed butyrate producers to produce butyrate. And that's, of course, where feed additives can play quite a role. You can steer this whole interaction. You can steer production of these beneficial metabolites, and then, of course, have the observed health effects. Just one final story, just a few minutes, just because some people were interested in this. It was not in the first slides, but if you think about butyrate and epithelial cells, they are different things. On the left side, you have transporters, and it means that if you have butyrate in the gut, the epithelial cells can transport it in an active way inside the epithelial cells. You have different transporters. This is, for example, a very important one, monocarboxylate transporter 1, but you also have some others. You also have transporters that pump it further to the blood, okay? This is not, well, you don't find it in high concentrations in the bloodstream. Butyrate can actually feed the TCA cycle, so it can lead to enhanced proliferation of epithelial cells, but it can also have an effect on an anti-inflammatory action because it goes into the nucleus and is involved in histone deacetylation without too many details. On the other end, you have quite some receptors, and you have two important receptors for butyrate, this one here and this one. And these are also involved in anti-inflammatory actions of this molecule, okay? And what I just want to point out here is that you have butyrate, okay? But you also have many other metabolites. And butyrate is a well-known one, but if you look at all the metabolites that bacteria produce, often these metabolites have effects on one of these transporters or on one of these receptors. That's amazingly interesting because, for example, if you have a lot of proteolytic degradation of 30 minutes. This is the first time I do this. I didn't realize it made such a noise, otherwise I would have done that. But just 1 minute ago. So if you have proteolytic degradation, so too many protein degradation, for example, in the gut, you see that you have a lot of ammoniac, and this is really blocking this transporter. So if you have too much proteolytic degradation there, you have less butyric acid uptake in epithelial cells. So that's why, for example, this ammonia in the gut is quite harmful. Same with hydrogen sulfide. Also here, too much proteolytic degradation of, for example, sulfur-containing amino acids. It blocks TCA cycle. And you can give you much more examples. Lactate, it can fuel, as I just said, it can fuel butyrate production by other bacteria, but it can also bind to some receptors present on immune cells. And what you see here, this, is like the receptors that respond to butyrate, they actually drive the immune system towards a lot of regulatory T cells, okay? This is also an anti-inflammatory cascade producing interleukin-10, okay? And basically, you can have a lot of different other compounds. Propionate is also affecting this butyrate receptor. You have vitamin B3, it's binding to this receptor that is also anti-inflammatory, and so on and so on. So all metabolites produced by bacteria, or at least a lot of these metabolites produced by bacteria, they, one or another, have an effect on one of the receptors, transporters, or systems that are responsive to butyric acid. So it's very complex, but I think butyrate is really a main metabolite, and all the others that are either produced by fermentation of amino acids or proteins or sugars, they actually play a role somewhere in this butyrate pathway, okay? And lucky you, this is the final one. As I said, butyrate is important. You can have a lot of strategies that enforce butyrate production, but it's much more complicated than that, and I think future will tell us how complicated this really is. So that's the final one. Thank you.
Filip, thank you for the exciting presentation, and I think there's a lot to think about. 13 questions, so we start with number 1 and finish with number 13, or?
I deliberately went over time to not to reply to it, but it seems, yeah.
No chance.
Yeah, whatever, maybe I take this one. I think, well, transcriptomics I wouldn't really focus on because it needs RNA, of course, and that's unstable, but I get the idea. Ideally, you shouldn't just look at species or genera or whatever, but you should look at genes that encode for microbial pathways. I think this is the future. Doing metagenomic studies, check which pathways of the bacteria are important, and then quantify these. I think that's basically very important because, for example, this bacterium, maybe other bacteria have exactly the same function, can produce exactly the same metabolites, and that's why probably we will need functional markers instead of just markers based on taxa or phylogeny. That's just one. One is enough, or should I do more? No.
One more.
Oh, one more. Damn. Okay. Okay. I don't think it's wrong to look at the microbia in the lumen because, well, I think the end aim is, of course, to look at microbiota in fecal material or colony content, because that's the easy way to do for the field. So I don't think it's wrong. And I also think that if you look at luminal content, this is something what we obviously always do quite often, you also see patterns that look very similar as what you see in the mucosa-associated community. It's different, but in terms of ratios between populations, it's actually, you can often see similarities. Also when you look at fecal material, which is obviously what we should do for field testing. Yeah.